Hi, good morning students. Now this class 12th standard biozoology, the unit number 2, chapter number 5, molecular genetics. The subtopics, now I am going to teach about human genome project, that is otherwise called HGP. In previous classes already explained the molecular genetics in different subtopic. Now, what is HGP? Human Genome Project. The total analysis of or analyzing the genome of an organism. The total composition of genome in an organism is called a Human Genome Project. What is called a genome? Genome means all sequence of the DNA or genes in an organism. This is called as genome. Genome are nothing but a group of genes in an organism. That is called as genome. Project means it is a program to analyze all the DNA sequence in an organism. That is Human Genome Project. The International Human Genome Project was launched in the year 1990s. The International Human Genome Project was launched in the year 1990s. It was a mega project. So it was completed, it took 13 years to complete. 13 years to complete. Why it is a mega project? It is a large project because one sequence, it takes more amount or more process. It has several years. So it took 13 years to complete. 13 years to complete. The HGB is 25 times larger. 25 times larger than genome of any other organism sequenced to date. 25 times larger than any other organism, sequencing of DNA in an organism. So approximately the total base pairs in organism is 3 into 10 to the power of 9 base pairs. So note it, approximately the Human Genome Project contains 3 into 10 to the power of 9 base pairs. So this project, it is a larger project. It is begin on 1990s and end with this end of the year is 2003. The HGP first started in the year is 1990s and completed this process or project in 2003. So it takes 13 years to complete. So HGP, first it is originated, this organization is US and National Institute of Health. So this is the total sequence of the DNA in human is the Human Genome Project. It is a mega project, 13 years to complete. It is a 25 times larger than any other sequence. Next one is it approximately contains 3 into 10 to the power of 9 base pairs. End of the year, this started in 1990s and completed in 2003. So it is organized by National Institute of Health in USA. So another important part of the Human Genome Project. Previous years, there is no software and developed computer programs. But recent years, it is a well-developed, rapidly development of the other technique to determine the sequencing of the DNA. Is very easy method. Because of available of many information on many computers to store the information about the DNA or genes. So particularly the HGP associated with the rapid development of new era in biology is called bioinformatics. Bioinformatics. When the bioinformatics is started, when it is developed after the rapid development of new technique. What is called bioinformatics? Collection of the information about the biology, biological information, particularly the biological information, DNA sequencing. So all the information related with the human, particularly this information is called bioinformatics. So bioinformatics is it leads to highly important field to development of the HGP. So next, what are the major goals for HGP or Human Genome Project? So now, what are the major goals for HGP? The first one is to identify all the genes in organism, particularly in human. To identify all the genes, approximately 30,000 genes in human DNA. How many genes are there? In one cell containing 30,000 genes are identified in human DNA. So this is one more question. So that through the HGP, how many genes are identified in your cells? 30,000 genes. How it is gather this information? Through the bioinformatics. So the scientists analyzed, isolated the chromosome, then isolates the chromosome and a DNA base pairs and identify number of the genes in a cell. That is 30,000 genes. The second important technique to determine the sequencing of 3 billion chemical base pairs. Is identify how many genes, totally 30,000 genes, 3 billion base pairs. Base pairs are in your cells. So that is 3 million, it determined the sequencing of 3 billion base pairs that make up a human DNA. It is another important goals. So all these two, one more question. How many genes identified in your cells? That is 30,000. How many billions of the base pairs or nucleotide chemical base pairs that make up a human DNA? Is that 3 billion base pairs are present. Next, this information is stored in databases. Store this information in databases. What is database? Data means what? Information. Collection of information is called a database. Collection of information is called as databases. Data means what? It is information, collecting the information. Which information? 30,000 genes, 3 billion base pairs, it is one of the information in databases. Another one is the improved tools. Improved tools for data analysis. So these are another goals. Improve the tools. Tools means what? Some of the materials or available information is called tools or chemicals, or it may be other enzymes are used for data analysis. The next one is called this HGP is related with other sectors such as industries. So the transfer the related sectors, other sector, related information, related techniques or information to other sectors. other sectors such as industries. Okay. So not only in biology, it is an industrial science. It is whole level, the total level of the information that are distributed, it's established, the world level is called such as industry level. Another important point, address the ethical, another one is legal and social issues. Social issues may arise from the project. That is ELS. Or it is ILC. LC. What is the expansion of LC? Ethical, legal and social issues. Ethical, E for ethical, L for legal, S for social, I for issues. It may be arise from this project. Ethical means what? Moral. That is, another one is legal means through the law. And the social issues may arise from the project. So I repeat, what are the major goals for HGP? It may ask 3 mark or 2 mark questions. Or even that is a 5 mark question. The first important point, through HGP, identify all the genes, that is 30,000 genes. The second point, to determine the sequence of 3 billion base pairs or chemical base pairs that make up a human DNA. And it helps us to store the information in databases as databases, to improve tools for data analysis. What are the requirements needed to the development of HGP? They are called tools. Next, transfer this information in world level such as industries. Next one is address the main thing for ethical, legal and social issue, that is LC. It may be arise from this project. Okay. The next, what are the methodologies of HGP? Methodologies of HGP. Methodologies of HGP. Methodologies of HGP involved, there are 2 approaches. 2 approaches. How many approaches? 2 approaches. One is the first approach. Identify all the genes. All the genes expressed as RNA. Expressed as RNA. That is, this is called as EST. EST. What is EST? Expressed sequence tags. Expressed means it expressed the genes, sequence means order, tags means it is files. So to identify all the genes expressed as RNA, this is called as first approach. What is the second approach regarding the HGP? Sequencing annotation. Sequencing annotation. What is called annotation? Annotation or the explanation of information. Explanation of information through the words, or it may be pair, or it may be diagrams. The clear information or note, any information. For example, this Human Genome Project, it is one of the explanation, in detail explanation through the notes is called annotation. So HGP is annotated data, is fully explained. So this is the sequencing annotation. Here, the second approach, this is the first approach, identify all the genes expressed as RNA, that is expressed sequence tags. The second approach is sequencing annotation, that is explanation of information. Here, the second approach is, it goes very details because it identifies the whole set of the genome. The second approach. To isolate the whole set of the genome. Whole set of genome. Whole set of genome. To identify all the genes that including coding and non-coding genes. Because in a DNA, there is a double helix DNA, which is made up of nucleotide sequence. Nucleotide sequence. For example, this end is 5N, this end is 3N. Here it is a 3N, next one is the 5N. So our DNA is made up of nucleotide sequences. Nucleotide sequences. The total number of the genes in a cell or an organism is called the genome, the whole set of the chromosome. Whole set of the chromosome. So the total set of the DNA is isolated. DNA is isolated. Through that, it cut into small fragments. For example, this is the long fragments. This is the long fragments. This end is 5N, another one is 3N, next one is 5N. This is made up of nucleotide sequence, for example, A, T, C, next is C, next is G, next is A, T, C, next is G. This is a nucleotide sequence of one strand. It is another strand. It is T, next to A, G, C, T, A, G, next to C. Now it is a base pairs nucleotide sequence, that is nitrogenous bases, pentose sugar and phosphoric acid. So a nucleotide is made up of pentose sugar, nitrogenous base and phosphoric acid or phosphate group. So that is a nitrogenous bases are two. One is purine, another one is pyrimidines. Adenine and guanine are purines. Cytosine and guanine or thymine are called pyrimidines. So first base pairs are purines, adenine and guanine. Pyrimidines are the thymine and cytosine. Thymine and cytosine. So here, how it is a base pair makeup? Adenine always combined with thymine. Guanine always combined with cytosine. But purines are adenine and guanine. Pyrimidines are thymine and cytosine. This one adenine is always combined with the cytosine by double hydrogen bond. Here double hydrogen bond. This is a triple hydrogen bond. 3. Here adenine always combined with the thymine. Cytosine always combined with the guanine. Because one purine combined with the one pyrimidines. So this is called a DNA base pairs. DNA base pairs. So how it is? The whole set of the DNA from a cell was isolated and cut these DNA fragments into small pieces. It may be cut into small pieces like this, small fragments. These small pieces by restriction endonuclease enzyme. With the help of restriction enzyme, cut that whole lengthy DNA strand into small fragments. These each fragments are smaller in size. Fragments means what? The total length of the DNA strand cut into small pieces. These are small fragments, small sized fragments. These small sized fragments, it must be cloned. What is cloning? Cloning means increase the total number of the copies. That is exact copies. So clone are nothing but exact copies of DNA. For example, here, how many fragments are there? 5 fragments is there. The 5 fragments are cloned into 10 fragments. So each fragments are same. This is called as cloning technique. Here, that is these 5 fragments are converted into random repeated fragments. It may be number of fragments. This is called as cloned. How it is possible? As a result of amplification of the DNA sequences. What is amplification? To production of multiple copies of DNA sequence is called amplification. Because only a single strand or it may be limited DNA base were not enough. It must be cut into small pieces. Small sized pieces that must be cloned. Again, that is amplification. Amplification means increase the number of copies, increase the number of fragments, multiple copies of DNA fragments by using host. That host acts as the vectors. Host acts as vectors. So commonly, the bacteria and yeast are used as vectors. Bacteria and yeast. What is called host or vectors? An organism carry the other DNA fragments. Other DNA fragments is called as vectors. So there are 2 important organisms are used. One is BAC, another one is YAC. BAC means what? Bacterial artificial chromosome. What is explanation of the BAC? Bacterial artificial chromosome. The second one is yeast artificial chromosomes. There are 2 organisms are used. One is bacteria, another one is yeast. These 2 organisms act as the vector that carry the DNA fragments, the number of multiple copies of DNA fragments. So this is important part, bacteria and yeast organism. So for this technique, to determine the total sequence of the genome of an organism are isolated by using certain enzymes and the chemicals are used. Physical and the maps which are used for determining the total sequence of the DNA or genome. These are called sequences. By using computers, by using automated DNA sequencers. Automated DNA sequencers. Automated using DNA sequence. This is otherwise called Sanger DNA sequencers. Because this was discovered by Frederick Sanger. So note it is an important one more question. The automatic DNA sequenators was discovered by Frederick Sanger. This otherwise called as Sanger DNA sequences. Here, the number of the copies of DNA genomes are produced. Certain genome are repeated sequences. Repeated sequences. The repeated sequence are called as microsatellites. Microsatellites. What is microsatellite? Repeated sequence of DNA. This is called as microsatellite. Here, this sequencer are used to identify the total number of the genome, total genomes. Here, nowadays the latest method, the total length of the genomes also identified. The total length of the genome, it may be longer It is not a cut into small fragments. See, this method is called shotgun sequence. Shotgun sequencing. What is shotgun sequencing? To identify the longer DNA sequence. Longer DNA sequence. This is a new method. So this shotgun method is a technique, latest technique to identify the entire DNA sequence. It is not fragments. Entire DNA sequences. This shotgun sequencing of DNA is replaced the traditional analysis of genome of an organism. So that is a two method. First one is an automated sequencer, was discovered by Frederick Sanger. The second latest method, recent method is shotgun method. What is shotgun method? It is one of the recent technique to determine or sequencing of longer or entire DNA sequences. Okay, so now what are the salient features of the HGP? Salient features of HGP. So the HGP is used to identify all the genes. Already I told you total number of the genes are 30,000. How many nucleotides base pairs are there? There are 3 billion base pairs. To identify 3 billion base pairs in organisms. The second goal for this. So total number of genome, average gene consists of, particularly that is a 3,000 genes are there. Sorry, 3,000 base pairs. Now the longest, that means the largest gene is dystrophin. Base pair, 3 billion base pairs are there. The largest gene are the dystrophin. What is a dystrophin? A protein present in skeletal muscle. Because it consists of 2.4 millions of the base pairs. So that is the largest gene, is a dystrophin. Third thing, the chromosome number 19 is the highest gene density. Highest gene density. And chromosome number 13 and Y chromosome are lowest gene density. So that is, I repeat, this is important. All these points are one more question. Chromosome, which among the 23 pairs of the chromosomes, which number of the chromosome have highest gene density is chromosomal number 19. Which chromosome are the lowest density is 13, chromosomal number 13 and Y chromosome. Similarly, how many genes, which one is the largest chromosome? That is chromosomal number 1, which contains 2,968 genes. Which chromosome have highest number of the chromosomes, highest number of the genes? Chromosomal number 1 among the 23 pairs, the first chromosome have more number of the genes. The totally it is 2,968 genes. Which one is the least number of the chromosomes? The chromosome Y. It contains only 231 genes. 231 genes. So listen carefully. Which gene was highest density? Chromosomal number 19 is highest gene density because it contains more number of the genes. Chromosome 13, because gene density means it is total numbers. Chromosomal 13 chromosome number 13 and Y are the lowest gene density. The total number of the genes present, that is 2968 gene is present in chromosomal number 1. That the Y chromosome is always shorter. This chromosome contains only 231 genes. So already I explained that microsatellites. So here 99.9% of the genes are exactly same in all the people. Same in all people. Or all person. 99.9% of the genes are similar in all persons. Only the remaining percent are repeated portion. Repeated DNA sequence. What is a repeated sequence? DNA sequence are called microsatellites. Tell what is it? Microsatellites. So that microsatellite are repeated, no direct code for proteins. Already I told you. Coding genes and non-coding genes. Coding genes are nothing but they contains information for protein synthesis. That is code for proteins. But non-coding is not for functioning of the genes. Here, the repeated portion, no direct effect are coded for proteins. But it is also very useful for analyze the structure and the genetic diversity. Genetic diversity means what? It is a more number of the differences which are present here. Differences. The next important point, that is over 50% of the discovered genes. 50% of the discovered genes are unknown. Discovered genes are unknown. Less than 2% are the discovered genes are useful for protein. Useful for the proteins. Because every genes are responsible for the synthesis of different type of the proteins. Okay, this point. Another important point, the repeated portion of the genes make larger. So the repeated portion are the polymorphic characters. That is called as SNPs or it is SNPs. Single nucleotide polymorphism, the single gene base pairs, the DNA differences. This is called what is it? SNPs, single nucleotide base pairs or single nucleotide polymorphism. Single nucleotide polymorphism. What is called a polymorphism? Yeah, multiple copies of DNA sequence. Multiple copies of the DNA sequence. So that is called as repeated portion. For example, CAG. These are repeated portion. How many times repeated the same course? 4 times repeated. 4 times repeated. That is a multiple copies. So that is it is always G. Next T, next C. The same. GTC. That is same sequences, repeated copies. More number of the copies. This one repeated again, 4 times repeated. So this is called as polymorphism. It is a repeated portion. So this can useful for identify the diseases related genes. To diagnose the disease related genes. So that is, it is cytosine always combined with the guanine, adenine always combined with the cytosine. This is a single nucleotide polymorphism. So one nucleotide sequences are repeated, so the repeated portion. So this is called as SNPs. This SNPs which is useful to understand the genes also, the disease related genes are identified in an organism. Okay, these are some of the important features about HGP, that is a Human Genome Project. Okay, that is identify 3 billion base pairs. The number of genes is 35,000 to 40,000 genes in a cell, in genomes. 35,000 to 40,000 genes are identified in HGP. So that is chromosomal number 19 is the highest gene density. Chromosomal number 13 is lowest gene density. Okay, these are the information about the Human Genome Project. So that is this subtopic is explained. One is what is the Human Genome Project, what are the major goals of HGP, what are the methodologies of the HGP, and what are the salient features of the HGP. Okay, so this is important topic in molecular genetics. So all the students must carry and take notes and gather the information and prepare well and save in your home. Thank you.